our panel of judges up here will rate each team on these 4 criteria with a score of 0 to 3, giving everybody a score of 12 points per team. I believe there are 8 judges, so approximately a score of 0 to 100. We will count up the scores after, and then we will announce the winners at the happy hour. Does that make sense to everybody? Yep. Yeah. Any questions on that? Scoring just became 1 to 5. So, it's about 160 points. So. A little bit. All right, so let's get started with some introductory remarks. So Onno, who is also doing Hackathon stuff, is also sort of running the podium for the presentation. So big round of applause for Onno. I think we got to speak to pretty much all of you during the hackathon. So if you haven't spoken to Onno yet, you can catch him during the happy hour. I mean, I'm sure he would be very excited to speak to you. Speaking to Onno is a lovely there. Yeah, I think the other people to really thank are Pete, who found me and really sort of logistically put this event together, and Laura, who is really the only organized person you see in the front of this room. So that's really it's been really nice watching her work, and she's really held everything together. And then I really had fun with all the judges, and actually I didn't know really any of these people before, but it was amazing to me. So Carlos was a little late, and he got stuck in traffic coming the first day. And the first thing he said to me was, "I'm going to get some water. Would you like some water too?" And I just thought that was such a nice way to meet somebody, like, so that was kind of cool. And in these hackathons in general, I mean, I think when I ran my first one, I told people it changed my life, and I think everyone changes my life a little bit, and this has been really special meeting all of you. I really mean that, and I hope it's been special for you too and changed your life just a little bit. And hopefully it changes the life of some people a lot. And we hope it changes science a little bit too. And I've seen some really cool scientific projects out there. And you guys really have been amazing because you come from non-homogeneous backgrounds, right? Like a lot of the hackathons I run, people are from very homogeneous computational biology backgrounds. You guys are all over the map. There are people that are professional computational biologists. There are people that walked in here and work on whatever you work on and learned enough about genomics to find something cool. There's one project that I think is particularly amazing in that sense. It's been an amazing experience. And Onno, did you want to say anything before we start?
Yeah, like since I get the opportunity, I don't know. It's been a really surreal weekend for me. Like I was sharing this earlier, like 3 very different days. I can say like Friday when we started, like a lot of excitement. There was really excitement that so many people showed up to work on this. Sharing my data, working on a real problem. I saw all the brain power in this room. It's quite impressive. So that was Friday. Then Saturday, I felt it was like a general high here. Like everybody was so like into it and working like crazy. And so my eyes were completely wide open the whole day. And I have to say it was hard to fall asleep after that day. It took me a while before the tiredness finally took over. And then today I feel a sense of everything got more realistic, right? Like people come up with real things, they identify genes that might be affected in me, or how to move forward. And you know, the talks and discussions, everything is like way more real and down to earth, which is kind of a good transition because I feel, oh my God, I wish this was over because I have to get down to work. There's so much to do. Yeah, so I want to thank everybody for showing up and yeah, together create this amazing weekend that I will not forget. Thank you.
That was amazing. So now we are going to move to the presentations. The first presentation is by a remote team called Team Edward. Awesome. So Team Edward, I was hoping they would send me 3 to 5. Are you guys here? Well, I mean, since you guys are up here, why don't you go ahead and present? All right, you have 4 minutes. Go.
All right. Right then. Good afternoon, everybody. We are Team Edward. My name is Kevin. This is Mustafa, Josh, and Ide, and our project is SNP and indel density cluster ranking. So we wanted to see if NF2 is driven by genes outside of NF2, and if the inactivation of this gene is what causes genomics and other diseases. So our goal was to locate the NF2 region and then measure the SNP and indel density in the NF2 gene and then find a pattern in the density. Then we use this to compare the genes without NF2 to see if they have similar characteristics to the pattern.
Great, so we retrieved the data from the FAR database and UCSC's H19 gene data. We retrieved the SNP and indel data using vectors from the VCF files. And for the testing data, we took Onno's tumor mutation data and subtracted Onno's brother's non-mutation data with ensemble. Thank you, Onno. Thank you, okay, so our methodology was to find the SNP and indel density in the testing and training data. And then we used a k-means clustering algorithm to characterize a gene by its distribution of SNP densities, SNP and indel densities. Okay, so given these density features, we created a cluster model. And we use the cluster model to identify genes with similar features.
So as for our current progress and the kind of results that we've come out with so far, so we're able to actually successfully train this k-means clustering algorithm to group these sequences that we're studying by the SNP and indel densities. And because we just started running out of time, we only attempted to study a small set of our entire test data. But we were able to calculate the SNP and indel densities for our test data and for our training data, but we just haven't been able to yet rank the test data that we're getting out. Yeah, we just didn't have enough compute time. We were literally just maybe half an hour out from finishing. But our biggest result was that we were able to successfully train this cluster model to recognize NF2 SNP density patterns. So to conclude, sorry, no worries. So to conclude, our original goal was to find other non-NF2 genes that cause the NF2 tumor to develop. We took existing NF2 variant data from ClinVar to train our data, to train a clustering model to identify genes that have similar SNP characteristics. And we attempted to use this model to understand non-NF2 genes. So we could use this tool in the future to identify SNP and indel characteristics to annotate genomes for studying other diseases or for like understanding and studying genes that may cause these NF2 tumors. So in the future, we want to use other metrics other than SNP and indel density to cluster data together. And maybe this might look like studying the SNP prevalence in the 1,000 Genomes Project or the allele frequency. We also want to study a larger data set. I know we were limited by time today. And we wanted to create a better fitting cluster model by varying our cluster parameters. And eventually we might want to explore integrating this with some kind of neural model. But yeah, we really want to thank you for listening to our presentation and for the wonderful opportunity of being here today.
We have time for one question. Are there any questions for this team?
Where's Edward?
I wanted to be Team Jacob.
I have a question. Any indications of, and you guys didn't finish the results, but any indications of any genes that have similar SNP and indel propensities?
So that's something we were just like right at the cusp of. Oh yeah, we were pretty much right on the cusp of figuring that out. Maybe I don't know. Maybe
Just out of your gut, did you see any like 3 or 4 other genes that popped up that kind of similar? No one's gonna help you for that.
Yeah, not yet. We're still getting results. So yeah.
Actually, there were some trends that we found. We concluded similar.
Can Jacob put them in?
It's actually, my name is actually Jacob.
All right. I would like to say about this team, they quietly worked. They were here late. They worked like crazy. Big round of applause for this team. All right, we got the exit stage left. So, Onno and I have decided to switch up the presentation order. So we're going to bring team Deep Drugs up now, and then after that, Onno will pick teams at random to present, which I think is a lot more fun. Deep Drugs. So I believe this was the team that spoke to me the least. So maybe that's why they were so successful. I don't know. They also seem to have more than 6 people, but you know.
We had a rotating lineup.
Oh.
Not everyone was here all the time.
Gotcha. So each of them was 86% of a team member. Hello, we are Deep Drugs. Okay. Who's presenting? Okay.
Hi everyone. Yeah, like you said, we are Team Deep Drugs. My name's Ryan. Well, this is my beautiful team. And we're here to talk to you today about what we spent our 3 days doing, and that was exploring novel and off-label therapeutics for NF2. So I'm a pharmaceutical engineer by training, so I was happy to bring into a project that I thought would leverage my biological background while also being able to pull on all the amazing AI and machine learning colleagues that I have around me. And so I don't think this issue really needs to be addressed. We know that NF2 does not have a currently existing targeted and effective therapeutic. And so our goal was to try and explore biologically mechanisms that we thought were of interest and that we could exploit towards developing future therapeutics for this disease. And so currently there's this large research database that's been growing towards the identification of druggable targets for NF2, but I think there's a lacking paradigm right now between the disease space and current existing therapeutics that really understand what these pathways are leading towards and what's actually causing this cancer phenotype to appear. And so our first goal was to really dig into the literature and find out what exactly is happening and what we think might be suitable drug targets. And so just based off these pictures, this is very simplified diagrams of some of the relevant pathways that we think are occurring in NF2. And from a biological nerd standpoint, NF2 is a really cool sort of mutation because it's a weird state of cancer where it not only affects extracellular receptors, but it also is interplaying with canonical pathways that are occurring inside the cell. And so you have this interplay between receptors that are feedback looping on the outside and then interplay from within the cell. And so when we were looking towards druggable targets, two things really stuck out to us. One of the canonical pathways from the extracellular receptors was an interplay with the inner receptors, and also there were finally targets that were not just general cancer targets that we often exploit, as you can see, like the mTOR pathway right next to the red box of targets that we're interested in. And so looking through this list of targets, we began a literature search looking for what we thought were interesting small molecule proteins that we thought could exploit some of these pathways towards inhibiting the development of NF2 into cancerous tumors. And so towards that, we utilized DeepChem and the macromolecular docking to then take each of our proteins of interest and then evaluate their binding affinity in relation to the ligands that we had selected. So going back to this pathway, looking at RAC1, CDC42 dual complex, the PAX, JNKs, and RAS complexes. So you leveraging DeepChem and its macromolecular docking platform to then evaluate the suitability and affinity of those pairs.
As part of getting DeepChem to work, we submitted 5 PRs and had them merged, and thanks Roth for merging them. So we were able to use the DeepChem web app to input various pairs of ligands and proteins and find their affinity scores.
We were encouraged when he told us not to use the GUI to in fact use the GUI. And so here's a sample run of us doing just that. I will say we spend an uncomfortable amount of time adding music to this video, so the fact that there's no sound is actually quite upsetting. And then yeah, so that last point is a readout on affinity scores for those pairs. And so we just want to highlight what we found was the most interesting. So this is CDC42, one of the markers that I was talking about, and then the drug target, the drug itself, AZA1, is a current explorative therapeutic. It was our favorite going in, just based off its biological mechanism, and it ended up being the strongest binding affinity in our macromolecular docking study. So that was a really positive result. Maybe some bias involved, maybe not. And this is just a proposed mechanism for what we think might be occurring when we look at what AZA1 is doing. So we think that through impediment of the RAC1 CDC42 complex, it impedes PAC1, and then pushes these tumor cells towards the cell survival, cell death mechanism, which forces them into apoptosis. So normally these cancer cells continue dividing, they continue progressing through the cell cycle without actually going into apoptosis. We're hoping that this mechanism recapitulates that and might show not only specificity for NF2, because we know that RAC CDC42 is not present in all forms of cancer, but also may have implications in other forms of neurological cancer. And yeah, future work, obviously, this was a very small subset of hand-curated molecules that we went through with a strong biological basis. I love the work that's going on with natural learning and language, be able to parse through thousands of articles and be able to pull targets. So doing something similar and then plugging into DeepChem would be great. Also, of course, I'm from the lab, so we'll have in vitro and in vivo studies in continuing development of models towards that, especially that might be able to leverage gene-specific and SNP-specific cell lines. And then, of course, continuing to engage the public and community through awesome events like this. I think, especially for rare disease spaces, knowledge is everything. So for someone like me who works in the industry and has never heard of NF2, now I have a special place in my heart for this disease. So I really do encourage events like these, continue growing, really continue reaching out to not only patients to make their data available, but also for people like us to continue engaging in the work. And so with that, thank you.
That was awesome. We have time for just one question. Anybody? Anybody? Going once, going twice. Rack does a lot of stuff, so what about side effects?
We will worry about side effects once we have efficacy.
Thank you very much. Another big round of applause for this team. They were awesome. All right. We exit stage left. Onno is going to pick the next team at random. We do not know whether that will be a remote or in-person team, so be ready. Hot dog, not hot dog? What a fantastic team name. From generally fun people. Generally. All right, 3, 2, 1, go.
Well, thank you everybody. We are the team H9H, which means hot dog, not hot dog, and at the very end you guys are going to see why we put landing. So I just want to say that this is a very diverse team and very much fun to work with. So I'm now recommending some also experimental biology and economics and this is a relational science protein person, and then Bob is a data science person. Mark is a machine learning person.
You know the microphone more to your face.
Yes, of course. So the approach that we follow, we wanted to set a baseline. The baseline was set up a baseline with the standard tumor normal analysis, and then based with our methods, then be able to compare against that to see what is the accuracy of our methods. So the standard tumor normal analysis was initially was the tumor, the normal, and the sibling, but we included a few other samples, but was mostly with those three. And then we sort of put the somatic variants from there. But the important part, we follow a regions of interest approach where we intersect two set of genes. One of them that was created with expression analysis, and then the second one with the list of genes that were created from high impact variants in the protein. And then at the very end, we sort of follow a variant filtering approach where we did interpretation of the variants. And at the end, we sort of did a few variant scoring algorithms. And then, of course, a big part of that was the regions of interest.
So I'm a developmental biologist, and I was really interested in looking at functional relationships of genes that are expressed at the same time and at the same space. So we use a data set where there's co-expression data and try to look for interactants with NF2 and interactants of those interactants, and try to merge those genes or the gene regions to the variants of interest. And from there we found, so we had high impact variants that intersect, so 107 high impact variants, and 4 of them intersected with the co-expressed gene list set. Some of them are enlisted above. Obviously, if we reduce the impact of the variants, we increase the number of co-expressed genes. So we stayed with these first 5 genes. The genes that we were interested in, and this is kind of the network, how related they are. So here's NF2, and this is one of the genes that it's associated with, and there are other 2 over there. So.
All right, so based on our analysis, we were interested in seeing how NF2 looked like, whether there were any mutations like stop codons, stuff like that. So out of our analysis, we picked 2 proteins, NF2 and EGFR. In NF2, we actually found 2 premature stop codons. And in EGFR, we found a bunch of structural interaction variant mutations, which is basically these mutations are synonymous mutations. However, they will affect the interactions of the polypeptide structure in the protein. These synonymous mutations that we find in EGFR2 might affect integration of the protein into the membrane, which can also have deleterious effects or enhancing effects. So next slide. I'm a structural biologist by training, so having seen these truncated mutation in NF2, we were able to look up a complex. So this is basically NF2 in complex with LATS1, which is a tumor suppressor gene. So the yellow part is Onno's version of Merlin, and the red part is the part that Onno's Merlin is missing. So this is the same complex with the part that Onno is missing. So as you can see, this truncated Merlin cannot interact with LATS1, which has a bunch of downstream effects. And this also opens the possibility as a structural biologist to look at LATS1 as a possible target, like maybe pathway inhibiting it or treating it in some other way.
Okay, so as you can see, the focus of our approach was to narrow down our targets for research. First, with looking at co-expression data to find probable expression targets that are possibly interacting with NF2. Our next thing we wanted to do after that was to rank genetic variants on mutations. We could find, we were working with the 1,000 genomes, and we cut it down basically to the co-expressed genes to make a smaller data set. And then as we were trying to develop a ranking model, we ran into two problems. We didn't have a large enough data set to work off of to train the model. And the second was, in place of that, our models were basically replicating already accepted statistical models based off of domain knowledge. What we ended up coming across, though, fortunately, is in Nature we found a paper that uses a boosted random forest that's been trained specifically to look for variants and rank them. And what's unique about it is it combines domain knowledge from the established measures that they're looking to improve upon. It gives us a base for our learning. And then what they added to it is about 298 features, but it's actually 3 separate vectors and 99 features plus 1 constant. But basically what they're looking at on there is looking at evolutionary constraints between aligned human, mammalian, and vertebrate to look for evolutionarily conserved things. And that helps to create basically an environment where machine learning can create its own knowledge on top of the existing domain knowledge. What we did with this is we used their pre-trained model. We couldn't compete with it. But it worked well for us. We were able to combine this with our reduced data set to come up with a set of targets to basically research, and then through their research on those, we were able to pinpoint the stuff we're looking at in the earlier slides. For future work, though, we would, with a larger data set, possibly for more NF2 patients, we could use a similar model to probably look at even more possible variant features beyond just what we're looking at here, and basically train our own model off of it.
And then, so this is the main idea that we have out of the hot dog, not hot dog idea. So we wanted to train the models and get the variants to say, yes, NF2, no NF2. So that was the initial idea where we're going to be with the clustering, and we're going to be able to say, based on a fixed set of variants, right, we're going to be able to say NF2, no NF2. Thank you.
That was fantastic. I think we're going to move along and not take questions. Most people get very intimidated when you stand very close to them. Obviously, this team is not bothered by such things. But can we get another round of applause? Thank you, guys. Please exit stage left and get a picture. Picture. That way. Yeah. Awesome. All right. So Onno's going to pick the next team. The reason I have my phone in my hand is because some of the remote teams have been sending bullet points on Slack. Aha! True cow.
Hi, my name is Chris. I'm one part of this amazingly diverse team right here. And today we worked on variant recalibration and improving that. Why are we here today? Well, ideally we're trying to take our expand our understanding of genomic data to translate that to improving patient care. And to do that, that really relies upon good processing of information as well as interpretation. You can see that errors anywhere along this pathway will result in downstream effects. And we've chosen to focus on improving variant call pipeline outputs because it's been shown that using different methods, you can get different results. Previous work has used unsupervised learning to help improve the quality of the outputs. However, with the recent release of the Genome in a Bottle truth sets, our goal was to use these truth sets as labeled data to then use a supervised learning model to help improve the positive predictive value of the outputs. So this is the approach. We have BAM data, which passes through a variant caller into a raw VCF, then it goes into our model, which has been trained using the Genome in a Bottle data to produce a true calibrated VCF. And this is important because all of you guys have been using Onno's data, the VCF files, and so our goal was really to improve your inputs. To tell you a little bit more about the methods, here's Alex.
Okay, so I was responsible for developing the models for doing this project. So I was essentially given the true VCF, and I was kind of told, okay, figure out what to put in a data matrix for this. And I'm like, okay, what do I do with this massive amount of data? So I tried to identify quality features while I was reading through the true VCF, and I identified the features which are detailed below. Each of these was explained to me in detail by Don, but I don't know very much about a lot of them. They're in the header file if you're interested. There was quite a bit of missing features due to the messiness of the data, so we imputed a lot of those features using the median of the features. You could also use other metrics quite easily. And then the labels, like Chris mentioned, were compiled using the Genome in a Bottle project, and this was just kind of a Boolean that identified if it was a real variant or not, so a true false field. So we used a very simple 3-layer feed-forward neural network in order to do this, and trained it using the Keras library in Python. This produces, in essence, a probability calculator, the probability being the result of the output layer, which is passed through the sigmoid or softmax activation, the final layer. So in essence, what we wanted to do was calculate the probability that the variant was a real variant versus a false positive. Kind of like we mentioned before, and the neural network drew upon other techniques which are mentioned in the slide. As for the results,
so unfortunately, we weren't able to pass Onno's data through this, but we did come out with a great model that we think incorporates these quality metrics of the genomic outputs. So it's on our GitHub, and we'd really love for you guys to use it, and we hope it's useful. Thank you.
That was incredibly complicated. Any questions? Okay, thank you guys. Do you mind if I send this to Justin Zook? And can he contact you? I'm absolutely serious. All right, great. Thank you very much. Please exit that way to get a picture. All right. Onno's gonna pick up the next thing. You know, so there's somebody like doing video of this, and she was asking me. She said, "Well, you know, I don't think I'm gonna film the whole thing." She said, "You know," and I said, "Well, you know what? You should get that team because that team's amazing, and that team's amazing, and that team's amazing, and that team," and then it just kept happening. It was ridiculous. So.
31.
Yeah. Deep drugs already lit? RNA-seq cluster. RNA-seq cluster. I know you guys are here. Yeah, that's you. Yeah, come on up. This is another extremely complicated project. And it is really, really amazing what you guys and the previous team were able to do in 2 days. What's that? Oh, you're missing something? What? Yes, you can go in 5 minutes. Okay. Here, guys, just hang out over there. You don't have to go all the way back to the back. Just there. Just, okay. Apparently, I have lost control of this whole situation. That's the secret. All right, this is definitely a remote presentation. No, don't skip it. I mean, we'll just start. We're still going to present the remote one. Skip. I actually have notes from somebody who sent me notes very early this morning. These are also on the Hackathon channel. So Parquet plus Avro is apparently more efficient for genomic analysis on Apache Spark than native bioinformatics formats, and often smaller on disk. So that's something I'm really excited about. That's awesome. And they use the Atom command line. I don't know if you guys have looked at Atom, but it's developed by some people at Berkeley and elsewhere. And so, yeah, so this is all available. This will all be available in S3 for you guys. And so this is showing you how small the other stuff was. Here are all the methods in Atom, which is pretty cool. Like I said, this will all be available in S3. They are also working on liftover to GRCH38, which makes me very happy. For those of you who don't know, GRCH38 has a lot of corrected errors as a reference genome, and also it has alt regions, which are often relevant. Here are some more methods, yeah. Here are some interactive genomic region joins in the Atom shell. So last, I would like to give credit to the people who actually produced this. Don't know why it's a slideshow. Oh, okay. I can't scroll down and see that. I'm going to bring up the GitHub page just so I can see the end of it. Hold on. So this is actually not the people that wrote this. These are, wait, there's two breweries. I have no idea what that means, but that's awesome. But I do know that this person is listening on Facebook, so can we please give these people, so we can I'm very curious about this. Can you put the link to the breweries in the Hackathon channel? Thank you. Pro team. And then we'll go back to RNA clustering next. Wait. Protein? Okay, back with. Yeah, we'll go back to RNA clustering. Okay, nobody leave unless you've already presented. Just hold it, please. Yeah, RNA clustering. Big presentation. Yeah. All right. 4 minutes, folks.
All right, hi everyone. I'm Akshay. This is Nikita. That's Dawn, Chitanya. We have Calvin and Paul. Today we'll be talking about predicting NF2 with data. Sorry about my voice. We had a lot of debate and arguments. Over the last 2 days, I lost a lot of it. So the question we want to ask is, what are the regulatory and gene expression changes that occur in NF2 disease? And second, can we leverage these to find new drug targets? That's how we started our discussion. Went on for a few hours. We then realized when we actually tried something that there's almost no NF2 patient data that's publicly available. No transcriptomic data, no epigenetic data, very little genomic data. I mean, we have Onno's data, but
N of 1 makes it hard to do any sort of bioinformatics or machine learning. So knowing that, we decided to go back to the basics. What do we know about NF2? What else can we look at to understand the regulatory networks and gene expression data to actually be able to do some bioinformatics and machine learning? All right, looks like an old version was put up. So,
yeah, so going, I'll just explain why we went back to the neural threads. So this slide here, we went back thinking about the basics, and NF2 comes from the embryonic cells of the nucleus. Shown on the left is a developing embryo, and those are migrating nucleus cells. And what happens is once the baby is born and comes these tissues, the nucleus gives rise to normal tissues as well as nucleus tumor. So what I want you guys to take away from this is these are the types of data sets we looked for and are publicly available. So we use NCBI SRA to go back and mine for RNA-seq data from early embryonic nucleus, adult nucleus, and nucleus tumor.
Thanks. Yeah, that's exactly right. So we decided to go back to neural crest data instead of only looking at NF2, because more broadly, we would expect that neural crest data has similarities with NF2 disease. So what we did was we used the NCBI database to look at different RNA-seq data, both from NF2 mutation diseases as well as any neural crest diseases. We then pulled all the RNA-seq data from these publicly available datasets, ran it through a bioinformatics pipeline to both do the alignment and the normalization. We ran it through some ML algorithms, classics like PCA or TSNE, and then we tried to identify which genes basically accounted the most for separation between classes, such as embryonic normal neural crest cells, neural crest tumors and cancers, and other NF2 gene mutations. So in particular, we chose about 5 classes, a couple from each of these, such as meningiomas and melanomas, and we tried to compare them to normal to see which genes could be used to most separate those classes, and try to identify those relationships with the goal of then being able to predict the outcomes and determine NF2 causing gene pathways and networks. So once we identified the genes, we're now at the step with things running. Our goal would then be to look at gene ontology of these top genes to determine NF2 related pathways that could then be used as potential candidate drug targets. Okay, so obviously we're having some difficulty with some slides, but in conclusion, we have a really awesome group here. It was amazing to see how diverse we were. So I'm a medical student, we have a bioinformaticist, we have a biologist, we have someone who's amazing at basically data security and optimization. We have 2 computer scientists, and with this group, we were able to basically collect 82 different data sets and put it through our pipeline in 12 hours, which is shocking to me. And yeah, I think we realized ML and genomics is something that can and definitely should happen. And with that, thank you.
That was incredible. Only a third of the teams have gone yet. I mean, the science that is coming out of this is absolutely amazing. So thank you, guys. Go take a picture. Go get a picture. That one. Yeah. Oh no. Do you want to pull up the next one? Yeah. So, Onno is so excited about people's presentations that he's not pulling up the next one, but that's okay. We are doing well here. And thank you to Onno NF2, who put the link to their slideshow right on the front of their GitHub readme. That was awesome. Isn't Onno NF2 a local team? Or no? No? Okay. Hopefully I have some notes. If the person who sent me the bullet points for Onno NF2, if your name is Bill, please say something in the Hackathon channel right now. So I got notes from somebody named Bill.
They're walking in right now.
Was that?
They're walking in right now.
They're walking in.
Right now.
Oh, where did you guys go? I thought you were local.
We are still local.
By the way, Bill, can you tell? That was amazing. You guys showed up right now. Come on up.
All right.
The perfect timing. Hey guys, welcome back. It's awesome that you guys came back here. What about the rest of you guys? Come on up. Yeah. Wow. The timing is incredible. Okay, cool. Here you go.
Hello, everyone. Neurofibromatosis type 2 is a very long known rare genetic disease, but it has an increased risk of other tumors, especially nervous system, including meningiomas. That is pretty detrimental to life. Because of it being rare and its rarity, there are limited resources that makes a challenge to identify novel therapeutic targets to further identify and understand the NF2 tumors. Additionally, understanding of the genomic aberrations or the mutations of this type of tumor is little known. Also, what makes the tumor good versus the bad or the differences is still really unknown. There are numerous clinical trials that have been performed, but have failed to identify systemic medical therapies that can effectively control the relentless growth of this tumor, perhaps due to the reason of its being rare. So one of the potential solutions for such rare tumors is to find bigger data sets or bigger sample size, which is it's almost impossible to do in today's time. So what is the potential solution? Would be to be able to play with the amount of data set we have now, and that's where the deep learning component comes into existence. And my friend here would describe the whole deep learning aspect.
So we propose a two-fold approach. The first approach, we use deep learning on NF2 data to train an autoencoder. And then we interpret that model to extract biological significance. The novelty of our idea is to leverage a larger data set and then train it on our smaller available data set to extract meaningful features. So we use autoencoders, which are a type of neural network that are used to extract features from very complex data. So the autoencoder is trained to regenerate the input, and it forces the network to generate meaningful encodings of complex data. So the weights contain very important information about what features are useful. So for example, if there's a large weight from node A to node B, then that implies that node A potentially influences the biological property underlying node B in a very significant manner. So we use transfer learning because the amount of data that we had to work with initially was very small. So we train on a larger NF1 data set, and then train on our NF2 data set to get meaningful features for the NF2 disease. So how we go about doing that is we analyze the weights for the encoding, and we identify nodes in we look at nodes in the hidden layer and look at the weights that go back to the input layer, which are genes and their expression, the expression data. We identify outliers based on the weights of these nodes and pick out genes that interact interact strongly with these nodes. And these hidden nodes could potentially represent biological features such as microRNA or transcription factors.
To highlight further the biological relevance of this exciting neural net is, as you can see, there are certain genes that have been highlighted by the neural net or are predicted by the neural net. These are all very well-known oncogenes, and in fact, they've also been very well studied for metastatic type of cancers, perhaps one of the major leads to really understand rare types of tumors. Also, we didn't stop there. We wanted to see how efficient is our neural net. And what we did was we took the neural net genes and performed a RNA-seq differential expression analysis. And you see here is the heat map, where your left is the normal cells or the neural crest cells, and the right is the NF2 tumors. And what we see is we could actually validate with the existing in silico technique that all these genes, such as EGFR, SOX9, ALA1A, etc., were significantly upregulated in the NF2 tumor types compared to the normal, which really it was exciting for us to see, and we were very happy that this is a significant observation coming out of a neural network that was tried for the first time. And we didn't want to stop there. Because we already had the neural net working, the question we wanted to ask is, can we really use this neural net to predict other biomarkers, perhaps circulating biomarkers, so a patient walks in the hospital and tells the symptoms, and the symptoms are not very clear, and the doctor can draw blood of patients and see whether these biomarkers are specific enough for NF2 tumor type, or perhaps be able to identify or monitor the progression of the disease. And what we found was that one of the microRNAs, the microRNA 200A, this neural net could show the association between this microRNA 200A with all the predicted genes coming out of the neural net, which also I should say this microRNA 200A is extremely well studied and also associated with poor survival outcomes in various tumor types. So quite exciting work. For future, first of all, I should say this is the first of a kind, almost a dream come true, because I had no idea deep learning could be applied in such a manner. And what we saw was mind-blowing based on all our observations. We could also leverage that a larger related data could effectively help us identify different targets as well as biomarkers. For example, for a smaller data set such as NF2 tumors, we could also train the neural net so that we can incorporate various other parameters to increase or better performance for the model. As I mentioned, we can identify potential therapeutic targets. Interestingly, what got me very excited to see was the EGFR in the neural net. And there are a lot of ongoing efforts and trials for EGFR inhibitor like alnotinib in various cancer types, including NF2 tumor. So it's kind of convincing that, okay, this is a great way to proceed further to understand rare diseases such as NF2 tumors. And also it has the potential to identify circulating microRNAs for early diagnostics and perhaps just to monitor the progression of the disease. And now coming to the most important slide is the true interdisciplinary team that we could create. Myself, Joe, and all of the team members here, we are very excited and we are very happy that we were given this opportunity to really go crazy with all kinds of scientific ideas. And please feel free to come to us and or email us and find us, and we would be very happy to go for further, deeper scientific discussions of our findings. Thank you very much.
That's how you walk into a room. So if you go out that way, he's going to take a picture of you and then cycle back around and come sit down, watch some of the other presentations. And I think there's a question real quick. Wait, guys.
I think I'll contribute this to DCAM. I think it would be very useful to have target tools for this. So yeah, let's chat about this. And in general, to everyone, if you have very cool tools you'd like to build that you'd like to get out there and get maintained, contribute it to DCAM, come find me afterwards.
Awesome, thanks. This is on GitHub, right? Like all the scripts and stuff? Yes, all right, amazing. So,
thank you.
Paranoid androids. Is that a local team or a remote team? All right, let's see if we can find what's going on with paranoid androids. It's an awesome team name, by the way. I guess they can hear me. So that's great. I am going to go through their slides. I don't think I have talking points for them. Why did they choose this track? They want an actionable solution. So they're looking at PTK2 expression and NF2. And they're trying a new computational approach to find it. I'm reading the slides too. Yeah, they're trying to intercept PTK2 and heat shock protein 90 to slow down or stop. Okay, cool. We've seen structure of Merlin. They have I'd like to point out that they have citations that they put in their presentation, which is awesome. Wow. Yeah, so they're looking at mesothelioma, which is really terrible for a lot of people. And this is how they came to HSP90 and PDK2. They're modeling target molecules as a graph and extending deep chem. That sounds awesome. And they did a bunch of stuff. And this is really great. And no, I mean, and they found preliminary FAK inhibitor drug candidates. So what are those drug candidates? So they did some stuff with Pymol, which is really cool. They don't put the candidates on here yet, but they should verify the efficacy of the discovered drugs. Hopefully they can hear us on Facebook. If you can't hear us on Facebook, if you can post the drugs that you discovered in the next 5 seconds, that would be awesome. I'm sorry, not on Facebook, on Slack. YouTube. That would be good.
We're streaming on YouTube.
What's that?
We're streaming on YouTube.
Oh, YouTube?
Yeah.
Oh, they're sharing on YouTube? We're sharing on YouTube. Oh, okay. Anyway, apparently we're live on YouTube and not Facebook. But then anyway, maybe we can hear from them later. So big round of applause, just in case they can't hear us on YouTube. That's awesome. Oh, great. I'm very curious as to what those inhibitors are. Read good. Ah, yes, the Read Good team. This is the most confident team of winning of all of the teams.
Whoa.
They have it together. So let's see what they have.
All right, thank you. That's a lot of confidence up front. So my name is Luke, here representing Team Read Good, joined by Lucky, Joe, and Akansh, and we're actually super impressed with everyone here. It was like, we're just surrounded by a lot of talented people, and it was actually really great to see. So, disclaimer from our friends at the FDA. So we mine the internet for academic sources and general news to find additional possible treatment options. So we were doing the NLP track. So we want to basically see, can you mine papers and discover all the treatment options that have been suggested, whether they've been researched or just speculated for NF2. And so that's actually what we did. And of course, the purpose is to provide NF2 patients like Onno with a wide variety of treatment options. So we're looking for treatment options that can help slow or stop tumor growth rather than just deal with the side effects or affect the symptoms. So we're looking at things that were really core to the reduction in tumor growth. So our approach is, again, we started with academic journals, peer-reviewed, as well as more speculative sources, so news, anecdotal reports, other things, and we fed it into a library called OpenIE. And what OpenIE does is basically give you subject-object relation tuples from text. So it says like, this causes this, or this treats that. And we did a lot of custom filtering on the results that are returned through OpenIE, and a lot of post-processing to try to increase the actual quality of those tuples, because there is like just about every sentence has some subject relation object tuple in it, and obviously we wanted the ones that were just treatment options or potential treatments. So going more deep into this, data sourcing, a lot of web crawling,
different like meta search engines. We did a lot of data cleaning. We had to take all these PDFs and then have them be something that can actually be ingested by some of these libraries. So we had to convert all of them to plain text. We did apply those algorithms, did a lot of additional filtering and everything. We also looked at doing topic modeling with Jensen and LDA, which produces some like ridiculous sentences like that. But it does contain all the relevant words that you will want to know for a particular treatment option. And then what we did is we took the candidates and then we basically filtered them down. Dr. Marco looked at the all of the results and helped us basically determine which of these were nonsensical and which were actual more robust treatment options. Anna looked as well. Thank you for that. So the results are available. So we have a searchable database that lists all the conditions, treatments, results, and the evidence. I don't think I can click through on here, but basically you can search for them, and it has things that are more